we're talking about the fact that carcinogenic agents damage DNA, and when they damage DNA, there are some key regulatory systems that can be damaged. One key regulatory system that can be damaged are proto-oncogenes, and when they're mutated, they become oncogenes, and they can then result in the formation of a tumor. Another key regulatory system that can be damaged are tumor suppressor genes. And what do tumor suppressor genes do? They regulate cell growth. And so this is the discussion that we now want to have, and that is that what are tumor suppressor genes and how could they result in tumor formation? So the key idea here is that tumor suppressor genes, they decrease, i.e. suppress, the risk of tumor formation. And the two classic examples are P53 and retinoblastoma. So let's take a moment to discuss each of these. Now, what does P53 do? It regulates the cell cycle. In particular, it regulates the G1 to S phase. And the way I think about P53 is sort of like a traffic cop. Let's take a whiteboard. Let's imagine that the cell would like to go from the G1 to the S phase. So before it can go to from G1 to S phase, it's got to sort of go past P53. All right, now, when it walks past P53, P53 is like a traffic cop, and it basically says, all right, show me your DNA. And when the cell shows P53 its DNA, there's two possibilities. If P53 looks at the DNA of the cell and sees that the DNA is mutated, then P53 will actually blow a molecular whistle, and it'll call in DNA repair enzymes and repair the DNA mutations so that before the cell goes into S phase, the DNA is repaired. That's one possibility. Another possibility is that P53 looks at the DNA of the cell and says, there is no way this cell is going to go into S phase because if this cell's got way too many mutations and we can't repair these mutations. So in that case, what would P53 do? It'll blow another molecular whistle and it'll call in a molecule called Bax. And guess what Bax does? Bax goes in and it destroys BCL2. So Bax goes in and it destroys BCL2. And you know what BCL2 is? BCL2 is the molecule that stabilizes the mitochondrial membrane so that cytochrome C doesn't leak out. So if you knock out BCL2, cytochrome C will actually leak out of the mitochondria, and when cytochrome C leaks out of the mitochondria, it will activate the caspases, which will then create apoptosis of the cell. And so P53 has the ability to induce apoptosis when there's severe DNA mutation in a cell. And again, the idea is you want that cell to undergo apoptosis because it's got so many DNA mutations that there's a risk that it could produce a cancer. All right, so let's read together. What do I say here? P53 regulates the cell cycle, in particular progression from the G1 to S phase. Now again, what does P53 do? In response to DNA damage, P53 upregulates DNA repair enzymes. All right. However, if repair is not possible, P53 induces apoptosis. And I've already explained how apoptosis would occur. Now, it's important to note that both copies of P53 must be knocked out for tumor formation. And the way I think about this is it's sort of like an intersection, right? These are traffic cops, and if one traffic cop goes to lunch, then the other traffic cop can sort of man the intersection and there wouldn't really be any accidents. But if both traffic cops are not at the intersection becomes chaotic and there's an increased risk for accidents. And so the only way to develop tumors is to knock out both copies of P53. And that's called Knudson's two-hit hypothesis. All right? That both copies of P53 must be knocked out for tumor formation is this notion that both copies must be knocked out is called Knudson's two-hit hypothesis. Now, loss of P53 is seen in more than 50% of cancers. Usually, both copies of P53 are knocked out somatically. However, it is possible that a patient could inherit one bad copy of P53 from a parent, and that's called the Li-Fraumeni syndrome. And the Li-Fraumeni syndrome is associated with an increased risk for multiple types of carcinomas and sarcomas. So that's a little bit about P53. Another important tumor suppressor gene is retinoblastoma. And retinoblastoma also regulates progression from G1 to S phase. And the idea here is that, I'll just do this as a picture, when the cell wants to go from G1 to S phase, one of the key molecules that it needs in order to do that is called E2F. So E2F is necessary to go from G1 to S phase, and guess who holds on to E2F? So E2F is normally bound by retinoblastoma. And in order for E2F to be released, retinoblastoma must be phosphorylated. And when retinoblastoma is phosphorylated, it becomes very happy, and it releases E2F, and the release of E2F then allows the cell to go from G1 to S phase. So that is the sort of the normal function of retinoblastoma. Now, if there's a mutation in retinoblastoma, then E2F is free, and the free E2F will allow cells to easily go from the G1 to the S phase, and that will then result in the formation of a tumor. So this is why loss of retinoblastoma increases or results in the formation of tumor. So let's go back and read. So retinoblastoma regulates progression from G1 to the S phase. Retinoblastoma holds the E2F transcription factor, which is necessary for a transition to the S phase. Normally, E2F is released when retinoblastoma is phosphorylated. And who phosphorylates retinoblastoma? Cyclin D CDK4 complex, which has the ability to phosphorylate proteins. If retinoblastoma is mutated, that results in constitutively free E2F. So for E2F will be free, and that allows for uncontrolled progression through the cell cycle. So that's the consequence of retinoblastoma mutation. Now again, same idea. Both copies of retinoblastoma must be knocked out for tumor formation, and that's called Knutson's two-hit hypothesis. If a patient has sporadic mutations resulting in both copies of retinoblastoma being knocked out, the patient will develop unilateral retinoblastoma. However, if the patient has germline mutation of retinoblastoma, the patient is more likely to develop bilateral retinoblastoma, and they also can develop osteosarcoma. Now, I think this requires a little bit of explanation, so I'm going to take a moment to back away and explain a principle here. Now, let's take, so what is retinoblastoma? Retinoblastoma is a tumor that develops in the back of the eye, and I've included a picture here in your text, and here's the eyeball, and this is here the tumor sort of in the back of the eye arising from the retina, and that's a retinoblastoma. Now let's take for a moment a very simple example. I'm going to go to my whiteboard here and just explain something. Let's pretend that there is a patient, and we're going to draw a couple copies of retinoblastoma. So this is these are normal retinoblastoma genes on their chromosomes, and retinoblastoma, one copy is inherited from mom and one copy is inherited from dad, and so this is a child and they've got two normal copies of retinoblastoma. Okay, now they've got, let's say in the back of the eye, they've got billions of cells sitting in the back of the eye, and each cell has two normal copies of retinoblastoma. Now, in order for a tumor to develop, one copy of retinoblastoma must be knocked out, right? And then in the exact same cell, that one cell in a billion, the second copy must also be knocked out. That's the only way, by knocking out both copies, that the tumor retinoblastoma is going to form. All right? So what's the probability that 1 in a billion cells would actually get hit, the exact same cell would get hit twice, and both copies would be knocked out? It's essentially 0. And that's why retinoblastoma is essentially almost never seen. It's a very, very rare tumor, and it's very unlikely that you would ever see it except on examinations. And so that's why I say here in your text that a sporadic mutation, the most those patients would ever develop is a unilateral retinoblastoma. Now let's take the other extreme, and that is, let's say that the patient has, I don't know, retinoblastoma, okay, here and here. These are the father and the mother, and let's pretend that one of the parents has a bad gene. We'll put an X through it. Now what's going to happen is that the child is going to inherit one bad gene and one good gene, correct? Let's just make that as an example, that they happen to inherit one bad gene and one good gene. Now, in the back of the eye, every single cell, all the 1 billion cells, just as an example, in the back of the eye are actually going to contain or already begin with one bad gene because they inherited one bad gene. Now, what's the probability that an additional mutation could come in and hit the good gene? Sorry, this is the good gene here. What's the probability that that could happen? It's pretty likely. It's not it wouldn't be unheard of. And so these patients, because the whole back of the eye is sort of like a field ready to be mutated, if any one of these cells gets hit, they're going to get a retinoblastoma. And similarly, if any one of these billions of cells in the back of the other eye get hit, they're going to get another retinoblastoma. And that's not too hard to occur because there's so many cells that can easily get mutated. And so that's why patients with a germline mutation have an increased risk of usually get bilateral retinoblastoma. And what's also important for examinations, there's an increased risk of osteosarcoma. So that's something to be familiar with as well. Okay, and the last or the third regulatory system are regulators of apoptosis. Now, remember that apoptosis needs to be regulated. And why does it need to be regulated? Because apoptosis, you want apoptosis to not occur in a normal cell. You don't want a normal cell to die, so you want to prevent apoptosis in a normal cell, but you want to promote apoptosis in mutated cells because you don't want cells whose DNA cannot be repaired. If there's DNA and it cannot be repaired, you want apoptosis to occur in that mutated cell. And so apoptosis is highly regulated. One of the important regulators of apoptosis is BCL2. Now, what does BCL2 do? I've told you this time and time again, but we'll repeat it one more time. BCL2 stabilizes the mitochondrial membrane so that cytochrome C cannot be released. If BCL2 gets disrupted, cytochrome C can leak out, which then activates caspases and subsequently results in apoptosis. So this is BCL2. Now, one of the high-yield principles to be familiar with regarding BCL2 is that BCL2 is overexpressed in follicular lymphoma. Okay, let's go back. Now, basically, the idea here is that in follicular lymphoma, you get something called a 14/18 translocation. Guess what sits on chromosome 14? Yep, immunoglobulin heavy chain. Okay, and guess what sits on chromosome 18? BCL2. You have to know that BCL2 sits on chromosome 18. Well, you would know that because of the 14/18 translocation. You know that BCL2 is involved in follicular lymphoma. So these are some things to be aware of. Now, this translocation, when BCL2 goes to the immunoglobulin heavy chain region, because we're dealing with B cells, this is occurring in B cells, then what's going to happen is BCL2 will be overexpressed. And then, because BCL2 is overexpressed, apoptosis will not occur because the mitochondrial membrane will be super stabilized because you've got super amounts of BCL2. Now, that's not really a problem because the vast majority of cells, and for the most part B cells, you don't want them to apoptose anyway. However, there is one region in the body where you actually want a lot of apoptosis, and that is in the follicle. Now, why do you want that? Because remember what's happening in the follicle. In the follicle, the B cells are undergoing somatic hypermutation. What's somatic hypermutation? That's a random generation of mutations within the B cell so that you can increase the antigen, the diversity of the antibodies being produced by the B cell. So somatic hypermutation is a desired state, is a desired occurrence within the lymph node, but if the mutations fail, you want those cells to die. That's the mechanism by which cells die. So we could state it otherwise. When B cells are born in the bone marrow, and they're born as naive B cells, and then they go to college. And where do they go to college? They go to college in the lymph node. And in the lymph node, when they go to college, the way by which they fail in college is somatic. The test in college is somatic hypermutation. Are you able to produce a newly mutated immunoglobulin that might be able to react against antigen. And if you're not able to do that, then you have to fail. And the way by which the lymph node fails the B cell and removes it is apoptosis. So if the B cell is not able to undergo apoptosis in the follicle, it's gonna just pile up. And as the B cells pile up, that's going to produce a follicular lymphoma. Okay? And so one of the key features of follicular lymphoma is that the cells are not apoptosing, and the reason they're not apoptosing is because there is a markedly increased BCL2, and so the cells can't apoptose because the mitochondrial membrane is super stabilized. So that's BCL2 and its relationship to follicular lymphoma. A few other important features concerning tumor development. We've sort of concluded our discussion of the key regulatory systems, but there are a few other features to be familiar with. First of all, telomerase is a necessary enzyme for cell immortality. Now let's go back and remind ourselves. First of all, cells, when they divide normally, each time a cell divides, its telomeres shorten. Okay? And telomeres are at the very end of the DNA of the of each chromosome, and the telomeres shorten, and every time they shorten, eventually that results in senescence. So the continuous shortening of the telomeres with serial cell divisions results in senescence, in which a cell basically shuts down and stops dividing. Cancer cells can't stop dividing. They need to keep dividing, keep dividing, keep dividing, and the cells are basically immortal. That's how cancer works. And so cancer often has upregulated telomerase, which preserves the telomeres so that cancer cells can avoid senescence. And that's an important general principle concerning cancer cells, that often telomerase is upregulated. Another important feature of cancer is that it requires angiogenesis. Tumor, if it's going to grow, is going to require blood. And so fibroblast growth factor, which is again an important angiogenic factor, despite the name fibroblast growth factor, and vascular endothelial growth factor are commonly produced by tumor cells to ensure tumor growth and survival. And then the final principle that I'd like to highlight is that tumor cells are mutated, correct? And so if they're mutated, they're going to be producing abnormal proteins. And if they're producing abnormal proteins, those abnormal proteins are going to be expressed on MHC class I. And one way by which the human body can deal with tumors is when there's abnormal protein present in MHC class I, the CD8 positive T cells come in and they can kill the tumor. And so CD8 positive T cells and MHC1 are highly important for something called immune surveillance of tumor cells. Okay? So one of the ways by which tumors can avoid this immune surveillance is by downregulating MHC1. If MHC1 is downregulated, the abnormal proteins being produced by the tumor won't be expressed on the surface, and the CD8 positive T cells won't be able to come in and kill those tumor cells. So that's one way by which tumor cells can avoid immune surveillance. Another important association here, or derivative principle here, is that when patients have immunodeficiency, that's going to increase the risk for cancer because immune surveillance won't be functioning properly, and immune surveillance is an important mechanism for removal of tumor cells, or for protecting a host from the development of tumor. But I would say very important to remember here that CD8 positive T cells play an important role in immune surveillance by recognizing aberrant protein present on MHC class I. And that really concludes, that's going to conclude this particular section. Relatively long section, but contains a lot of high yield. And so we spent a little bit of time here, but hopefully it'll pay off as we move through the course. So we'll go ahead and stop here, and the next section will be a discussion on how tumors can progress once they develop the necessary mutations to actually form a tumor.